Good day, everyone, especially to our dear panelists. Today, we will be presenting research from 2012 entitled as Phytochemical Screening and Identification of Some Compounds from Mallow. But before we proceed to the proper presentation, we would just like to introduce ourselves first. We are the presenters that will be presenting for this study. I'm Eowenens Valdez.
I'm Muhammad Marjan Ansano.
And I'm Joel Granados, and let us proceed to the introduction part of the research.
For the statement of the problem, the secondary metabolites are chemical classes that have been shown to have therapeutic properties against a variety of human disorders, which may explain why traditional medicinal plants are used to cure certain illnesses.
The aim of this work is to carry out a phytochemical screening of some extracts of Malva sylvestris L, seeds and stems in order to know the composition of secondary metabolites in relation with the structure of their phenolic compounds and better understand the pharmacodynamic properties of its extracts.
Moving on, for the null hypothesis, researchers hypothesized that the extracts of Malva sylvestris L seeds and stems and the composition of secondary metabolites have no relation with the structure of its phenolic compounds.
For the alternative hypothesis, researchers hypothesized that the extracts of Malva sylvestris L seeds and stems and the composition of secondary metabolites have almost, if not the same structure of their phenolic compounds. Many of the seeds and stems of mallow have tested strongly positive or positive on all three extracts used.
Now, for the significance and scope of the research study, the researcher chose to focus on carrying out the phytochemical screening and identifying of some compounds from a species of mallow known as Malva sylvestris L, in order to know the composition of secondary metabolites in relation with the structure of their phenolic compounds and better understand the pharmacodynamic properties of its extracts.
Now let's move on to the methodology. The researcher starts with a sample extraction. The triphytochemical test of seed and stem will be analyzed after extraction of three solvents: etheric, ethanolic, and aqueous. Then move on towards the phytochemical screening, in which there are 9 submethods, which are tests needed to see which compounds are present, namely phenolic compounds, saponins, alkaloids, anthocyanosids, anthracenoids, imidals, coumarin, sterols, steroids, and carbohydrates, and TLC, or thin layer chromatography, which will be performed on the silica gel plate. 2 microliters of each extract and standards were deposited.
Moving on to results and discussion. According to the triphytochemical screening of the extract studied of the seed in Table 1 revealed that alkaloids are present in large amounts with the reducing sugars, sterols, and steroids. Other classes are present in small quantities like tannins, imidals, starch, and coumarins. However, the seed extracts tested negative for the presence of anthracenoids, anthocyanosids, saponins, and flavonoid classes.
On the other hand, triphytochemical screening showed that the stem contains a higher amount of flavonoids and stanins, starch, saponins, alkaloids, imidals, sterols and steroids, reducing compounds, coumarins, anthocyanosids are present in small quantities. However, anthracenosis classes are absent in the mallow stem extract.
According to the phytochemical analysis of Malva sylvestris L, showed the presence of sterols, and the presence of terpenes is well noted. By cons, noted that Malva sylvestris L contained scopolamine. It's also noticed that tannins, saponins, flavonoids, and carbohydrates were found in the extracts of malva leaves. Literature reference about phytochemical screening of seed and stem parts were not found.
Separation of the bioactive compounds from two parts of plant extracts was carried out using thin layer chromatography, also known as TLC. The TLC analysis UV light allowed the identification of a pattern of phenolic compounds or blue fluorescent.
For both seed and stem, we noted the presence of two phenolic acids, which is syringic acid and paracumaric acid, by the first solvent system, and no compound is identified by the second solvent system as shown in the table. Work revealed 11 phenolic compounds that were isolated from water leaf extract of Malva sylvestris L. The main phenolic acids identified are gallic, pyrogallol, vanillic, synergic, cinnamic, and trypsin acid.
Lastly, we have the conclusion and recommendations. The seed and stem of Malva sylvestris L were used in this investigation contained a variety of chemical groupings. It discovered some differences in the components of the two plant sections examined. Seed extracts proved positive for all except anthracyanosids, anthocyanosids, saponins, and flavonoids, whereas stem extracts tested positive for all but anthracyanosids. TLC was used to identify syringic and parachromatic acid in both sections of the plant.
Given the therapeutic and medicinal capabilities of seed and stem extracts, it would be worthwhile to isolate and identify distinct compounds, then test them in vitro and in vivo using animal models to narrow down this therapeutic use.
Here are the list of the references used in our PowerPoint presentation. Once again, we are the presenters of the research phytochemical screening and identification of some compounds from mallow. I am Joel Granados.
I'm Eowyn Valdez.
And I'm Muhammad Marjan Ansano. Thank you for listening and good day for our fellow classmates and for our dear panelists.